In today's lesson, we are going to continue our Form 5 biology revision. Let's go! What is up guys? Welcome back to the video. Today we are going to continue on our Form 5 biology total revision. Before we begin, the PDF will be as the last video provided for free. Go to the links in the description and download it, and let's begin. Alright, so let's begin with our Chapter 1 Transport. Let's look at why do multicellular organisms like us need a transport system, whereas unicellular organisms like paramecium, they don't need a transport system. Okay, the keyword here is the large total surface area over volume ratio. I just put it short as TSA over V. This TSA over V ratio, if it is very big, you are like a bacteria, means you don't need a system. If it is very small, like us humans, means our surface area is not enough to support the volumes of cells inside of us. Therefore, unicellular organisms, they have large total surface area over volume ratio. Not only that, they have short distance between external environment and the cell, means from the external environment to diffuse into the cell, it's only a few at most. Therefore, simple diffusion is sufficient. They do not need a circulatory system. Whereas multicellular organisms, we have very small TSA over V ratio, large distance between external environment and cell, therefore we require a circulatory system. So, to make a circulatory system, you need three components. You need a pump, your heart, you need pipes, your blood vessels, and of course you need medium, which is your blood or hemolymph in insects. Moving on, we have a few types of circulatory system. Number one, it's the open system, where the fluid doesn't necessarily all the time be in the circulatory system. For example, insect, the body cells, they bathe in the hemolymph, means they will go into the pipe, they will go out to the cell, bathe it, and then they will go back in again. So it's not a very efficient system, but whereas complex organisms, we need a closed system, meaning blood is always in the vessel. Okay, we have a single system in fish, which means the blood passes through the heart once. Double system, which means the blood passes through the heart twice. Incomplete means there is mixing of deoxygenated and oxygenated blood, making it not very efficient. Complete means there's a separation between oxygenated and deoxygenated blood, making it very efficient. For example, in mammals. Next, moving on. In single system, there's only one atrium and one ventricle. Amphibians have 2A and 1V, remember? This one ventricle is what causing the mixing of blood. Mammals is 2A and 2V. The separated ventricle means there's no mixing of blood. As I mentioned just now, blood passes through the heart once. There is mixing of blood here, pass through the heart twice. Why is it twice? Because we have two circulation here. Number one, to go to the lungs, it's called pulmonary circulation. Next, to go to the body, it's called systemic circulation. The same goes for mammals. Moving on, human circulatory system, the blood has three functions. Number one, to transport oxygen and glucose to cell, and of course to take carbon dioxide and waste away from the cell. That's the most important function. Number two, it is to regulate pH, water, and temperature. Number three, it is to protect you from blood loss and of course from infection. Let's look at our composition of blood. 55% the watery yellowish part, it's called plasma. What's inside them? You have water, ions, salt, plasma proteins, for clotting and antibody, hormones, dissolved substances like nutrients and gases. All the line stuff will be in your plasma. Next, you have your blood cells, which makes up 45% of the blood. You have your red blood cell, white blood cell, and platelets. I'm sure you know what the function for each of them. Red blood cell has hemoglobin to carry oxygen. They have biconcave disc shape. White blood cell, you have two types here. You have granulocyte, which I call it the low-ranking white blood cell. The focus here would be your neutrophil. Neutrophil will carry out phagocytosis, means they will engulf bacteria one by one. Not a very efficient way to kill bacteria, but they have large number, means they can consume a lot of bacteria. Next, you have your agranulocyte, which I like to call this the higher class white blood cell, because you have your monocyte here, they can engulf bacteria much better. And of course, the best being your lymphocyte. They will produce antibody and then they will kill pathogen in a very efficient way. Or I like to say, lymphocyte is like the sniper here. Very accurate and very efficient. Platelets, they only do blood clotting, which we will go into that later. Moving on, we have your blood vessel. Arteries, since they have very large pressure, they have thick and muscular and elastic wall. And of course, they have very small lumen, meaning the blood is flowing very fast. Next, they will go into the blood capillaries. This has the largest total surface area because this is where all the substance exchange will happen. And then they will converge into your vein, which returns to the heart. Vein is thinner, less muscular, and of course it has bigger lumen. Since they have low pressure, they have blood flowing back problem. Therefore, you need valves along the way to stop the blood from flowing back. Moving on, this is the most important part. That would be your heart. I don't know why is the heart square here, because square is just easier to understand. Heart has 4 chambers and 4 pipes. So you have the right atrium, right ventricle, left atrium, left ventricle, and you have 1 pipe for each room. Right atrium has vena cava, right ventricle has pulmonary artery. Pulmonary means lungs, means it's going to the lungs. And why is it called artery? Because it's going away from the heart. Artery doesn't necessarily mean it carries oxygenated blood. It means it is carrying blood away from the heart. Away artery, away artery. This is how I memorize. And then you have pulmonary vein coming back. And then you have aorta carrying out from the left ventricle. And then you have 3 types of valves here. You have tricuspid valve over here, bicuspid valve over here, and then you have semilunar valve over here. So next, let's look at the point. Heart is made up of a cardiac muscle. You only have this muscle in your heart, and it is myogenic, means it beats by itself without nerve impulse. So even though your nervous system shuts down, your heart still beats. So how does a heart beat? You start everything with the SA node. This is where all the electrical impulse starts. Then this will cause the atria to contract, or we call it systole. And then this SA node will pass the signal to the AV node, passing it down to the bundle of His, or this Purkinje fiber. And then next, your ventricle will contract, meaning that atria will contract first, followed by your ventricle. That's why your heart moves like this. They take turns to contract. Moving on, why is the left ventricle thicker than the right ventricle? Very common in exam. To generate higher pressure to pump blood to all parts of the body. Moving on, let's go into our blood clotting system. What is the function of blood clotting? This is to prevent blood loss, to maintain pressure. Very important, if not you will die. And of course to maintain a closed system so that microorganism doesn't go into our bloodstream. So you have everything summarized here. When you have a damaged blood vessel, you will send out clotting factor. This is the signal that tells your body you are bleeding. So this will cause your platelet to clump together to form something called a platelet plug. But this platelet plug is not very strong. Therefore, you have this system, which is the most common in exam. This clotting factor will trigger a protein called thromboplastin, and combined with calcium ion and vitamin K, they will stimulate prothrombin to convert into thrombin, and thrombin will stimulate fibrinogen to convert into fibrin. This fibrin is like a mesh. It's like a web that will hold this platelet plug together. Think of it this way, platelet plug is like bricks blocking a wall, but not only you need bricks, you need mortar, you need cement. Fibrin is sort of like a cement that holds the bricks together and make the wall stronger. If it makes any sense. Moving on. So blood clotting problem, you have hemophilia, means they lack certain clotting factors, they cannot clot their blood. This is a genetic disease. And of course thrombosis, this is when you have blood clot inside your blood vessel. This is the most common cause for heart attack and stroke. Moving on, your lymphatic system, very important. So you have your blood capillary here. This is the artery end, it has high pressure. This is the vein end, it has low pressure. So due to the high pressure, plasma will be forced out into your cells, forming your interstitial fluid. Can you see this positive 100 here? And then at the vein end where you have low pressure, all this interstitial fluid will be drained back into your blood vessel, but not all, only like 75%. So you have like around 15 to 25% of interstitial fluid here, and if this thing accumulate, you can start to swell. So we can't have that. That's why we have our lymphatic system. This sort of acts like a recycling system. It collects all the interstitial fluid, turning it into lymph and return it to our vena cava. So how do they drain? Upper and right part of the body, they will drain into your right lymphatic duct. I would focus on this. And the left and lower part of the body will drain to your thoracic duct. And of course, at the end, they will drain to the right subclavian vein and the left subclavian vein. And of course, at last, they will rejoin everything at the vena cava so that your blood doesn't get less and less watery. It remains concentration. So you have something called lymph nodes along the way. This is sort of like a checkpoint because pathogens, they really like to go into our lymphatic system because it's full of nutrients. This is why you have roadblocks, you have police. These police are our lymphocytes. They will produce antibody and they will do immune response to kill all these pesky pathogens. You have your body defense mechanism. This is, I would say, one of the most important part of this chapter. You have your first line non-specific, second line non-specific, third line specific, or I like to call this the immune response. First line is like a Great Wall of China. It's a physical barrier. So you have your skin, it produces sweat and sebum, contains lysozyme, which is the enzyme that will kill pathogen. Mucous membrane contains lysozyme as well. Tears and saliva, hydrochloric acid in your stomach, and of course our blood clotting system. Second line is your neutrophil. The low class police will carry out phagocytosis to eat all this pathogen. Not very efficient, but they win in terms of numbers. Last but not least, you have the strongest guy. This is like the sniper, the SWAT team in your body. That's your lymphocyte. They will produce antibodies to the specific antigen on the pathogen, meaning that they will only target a specific pathogen. Lymphocyte will remember any disease you have gotten before. How antibodies work, this part I wouldn't want you to focus. 4 ways antibodies work. Agglutination means they stick all these pathogens together. Think glue, agglutination glue. Neutralization is to neutralize the toxins produced by the pathogen. Opsonization acts as a marker. These antibodies will mark the pathogens so that it's easier for neutrophil to engulf them. And of course, last, lysis. I said this in the last video. Lysis means to burst. It will make the pathogen burst so that they die. Next, you have your immunity. You have your active immunity, means you produce your own antibody. Passive means you get it from an external source. The quickest way to memorize this is to memorize example. Natural active means you recover from a disease. Let's say you get COVID-19 today, you recover. This is how you get COVID-19 immunity. Artificial active is when you go and get vaccine. This is what we are trying to do now. So why not get natural active? Because not everyone can survive natural active. Would you go and get smallpox? Would you go and get tuberculosis? You might not survive it. This is why we take artificial active, which is our vaccine. Natural passive, you only 2 sources here. One is the placenta, number 2 is the breast milk, all coming from our mother. Last but not least, artificial passive. This is what you get from an anti-serum. You get this from horses. They put the anti-serum into you, they put antibody into you. Moving on. Vascular tissue in plants, you only need to know where's the xylem. Always remember, xylem is always on the inside of the plant. Leaf, it is on the inside because the stomata are on the lower part of the leaves. So xylem is hiding away from it to reduce water loss. And then stem, it is hiding inside as well. Root, it is hiding inside as well. Really simple stuff to memorize. So transpiration, you have 4 factors affecting it. Light intensity, more light, more transpiration. Temperature, hotter it is, more transpiration. Humidity, more humid, less transpiration. Air movement, more wind, more transpiration. This is your transpiration. You have 3 things happening here. Inside your root, you have something called root pressure. When your root hair cell pumps salt, active transport into them, they turn very salty, so water will move in via osmosis. This is your root pressure. Next, moving up the stem, you have something called capillary action. This is caused by the adhesion force and cohesion force of the water column. Adhesion means they stick to the wall of xylem. Cohesion means they stick between themselves. Next, leaf, they will do transpirational pull, means water, they transpire away to the surrounding, means they have to pull more water into leaves. This is what we call transpirational pull. 3 ways to move water from the soil all the way up to a plant. Let's go into our chapter 2, locomotion and support. I wouldn't want to focus a lot on this chapter. Why? Because It is not very common in exam. So, 3 types of skeleton. You have your exoskeleton, the outside skeleton, insect, endoskeleton, inside mammals, hydrostatic skeleton means water, like earthworm. Human skeleton has 2 parts. Axial means the center part. You have your skull, vertebral column, your backbone, ribs and sternum, because these 2 are attached to your vertebral column. Appendicular means side, which is your pectoral and pelvic girdle, your shoulder and hip support, and of course your limbs, your arms and your legs. So vertebral column is 7, 12, 5, 5, 4, means you have 4 cervical vertebrae, the neck vertebrae, 12 thoracic vertebrae means your chest vertebrae, 5 lumbar vertebrae means your lower back vertebrae. Sacrum coccyx used to be our tail because we evolved from chimpanzee. Now we don't have it anymore. So your sacrum and coccyx just kind of sits there doing nothing. So you have your intervertebral disc to absorb shock. You have a donut thing sitting in between each vertebrae. This is to absorb your shock in the vertebral column. So let's look at how your vertebra looks like. So this is a typical vertebra. So a simple way to memorize here is, let's look at the note first. Foramen, whenever you see the word foramen, it means hole. Whenever you see the word process, it means a stick sticking out. When you see vertebral or spinous, means it is at the center. When you see transverse, means it is at the side. So when you see spinous process, it means center stick. A thing that sticks out in the middle. So this is a spinous process. And then you have your transverse process, means the side stick. So what are these sticks for? This is for the attachment of muscles. Next, you have your vertebral foramen, means center hole, and transverse foramen means side hole. What is this for? You can write this down. This is for the passing of your nerves and blood vessels. And take note here, transverse foramen, only cervical has this, because why? Cervical, you need blood vessels going to your brain. Others, you don't need that. And of course, this centrum here, this is for the disc to stick to. Very common in exam. Your lumbar vertebrae has a large centrum here. Why? Because your lower back bears the most weight of the body. That's why they have the largest centrum and the largest intervertebral disc. Next, you have your joints and antagonistic muscle. I don't really want to talk too much about this because this is very straightforward. Your joint, you have 2 types. You have hinged joint, it's like your door, and then your ball and socket joint, which is like a ball and a socket. This is the most flexible joint. Moving on, you have your joint, you have your bone here, you have your cartilage. This is to reduce friction. And your synovial fluid is to provide lubrication. They are like your oil. And then you have your synovial membrane here. Forgot to label here, you can add this on. Something is missing here, you can add this here. You have something called ligament. Tendon is muscle to bone. Ligament is bone to bone. Okay, moving on, you have your human arm here. Antagonistic means they work in opposite. When one contracts, one relaxes. When one relaxes, one contracts. Same goes for animals. This is in earthworm. When the circular muscle contract, the longitudinal muscle relax, they will become longer. And then when circular muscle relax, longitudinal muscle contract, they will pull the worm shorter and fatter. Next, you have your grasshopper. This is the hind leg. When this muscle contracts, when this muscle relaxes, they will fold their leg. And then when this muscle relaxes, when this muscle contracts, they will extend their leg. This is how they jump. So adaptation of fish, you have streamlined body, overlapping scale facing backward, you have slimy coating. All this is to reduce water resistance so that they can swim very fast in water. Let's look at the fins. This is also very important. So your pelvic and pectoral fin is to stop the fish from doing pitching. Dorsal and ventral fin, this is to stop the fish from moving sideways and rolling. We call it yawing and rolling. And of course, this caudal fin is to provide thrust, means to push the fish forward. You learn thrust in physics. Next, you have your bird. Bird has 2 muscles. You have the pec major, means the big one, and pec minor. In upstroke, since upstroke is not that important, the small muscle will do the work, which means the minor will contract, the major will relax. And then downstroke is where everything is important, because downstroke will cause the bird to go higher. This is how they fly. This is why they need the big muscle when they do downstroke. So the major will contract and the minor will relax. Moving on, let's go into chapter 3. Very important chapter, especially the kidney part and the homeostasis part. So your human nervous system has two parts, which is the central nervous system, the middle part, your brain and spinal cord, the most important one, and your peripheral nervous system, your cranial nerve and your spinal nerve. If you break your PNS, it is not that serious as breaking your CNS because you can die. So your CNS has a few parts. You have your cerebrum, which does voluntary action, things that you can control. And then cerebellum, a lot of students will make this mistake. They think, oh, cerebrum is voluntary action, that means cerebellum must be involuntary. No, this is for balance and muscle coordination. So who does involuntary? That would be your medulla oblongata and your spinal cord. Next, you have your hypothalamus. This is where your sensors are located. They will detect your blood pressure change, body temperature change, osmotic pressure change, and so on. So they will detect internal and environmental changes and they will do homeostasis. Next, pituitary gland. This is the master gland. They will control the secretion of hormones of all the other glands. Moving on. So you have three types of neurons here. You have your sensory neuron, which means to carry impulse into your body. Interneuron or relay neuron, this is to connect them. And then you have your motor neuron, means to send nerve impulse to your effector, which is your muscle. One thing very important here, since this is carrying it in, so you have long dendron, because dendron is carrying it in. And of course this one has long axon, because this is exiting the body. Think exit, axon, exit. Moving on, you have your synapse. This part, please focus, because this one they will ask it in essay. Make sure you know where are they labeled. So how do they work? Electrical impulse, it arrives at the axon. So this is the axon. Synaptic vesicle, which means these bags, they will fuse with the presynaptic membrane. Pre means before, which is this guy. And then they will release neurotransmitter, which is the chemical, across the synaptic cleft, which is this gap here. And then this neurotransmitter will bind to the receptor here on the postsynaptic membrane. Post means after. So this one will generate a new impulse and the impulse will carry on, and then they will kill this impulse at the axon. Please remember all these points because this is how you score in the essay. Moving on, let's go into reflex action or reflex arc. Okay, real simple stuff here. When you touch something painful or hot, your afferent neuron or your sensory neuron will carry impulse to your spinal cord, not your medulla or your brain. And then your relay neuron will send it to your motor neuron, and then it will send it to your effector, and then you withdraw yourself from danger. This is a very fast response in around 0.1 second, faster than you can think. Moving on, you have your hormones here. Pituitary gland will secrete growth hormone, thyroid stimulating hormone, luteinizing hormone, and follicle stimulating hormone. These two, we will go into that in our reproduction later. So it controls the secretion of hormones from other glands. Of course, you need to know GH, which is your growth hormone. Too much, it will cause gigantism. Too little, it will cause dwarfism. Very common sense. Thyroid gland, this is to secrete thyroxine because this will control your metabolism. Adrenal gland will secrete adrenaline. This is for your fight or flight response. It will make your heartbeat increase, your breathing rate increase, more glucose shipped into your cells. It will make you run faster. This is to avoid from danger as well. Pancreas, insulin and glucagon. Insulin, this is for diabetic patient, which means to convert glucose to glycogen to store sugar. And then glucagon is to release sugar to convert glycogen into glucose. Ovary, we will go into this in reproduction later. Progesterone and estrogen, this is for menstrual cycle and for your puberty, secondary sexual characteristics. Testes, same thing, your testosterone, they will make your sperm develop puberty in males. Next, let's look at kidney, your nephron. This is really important. Starting from your glomerulus, you have your afferent arteriole going in and your efferent arteriole coming out. Can you see the diameter here is bigger than this? Why? Diameter of afferent is bigger than efferent. This is to generate high pressure so that you can force all the rubbish out so that you can filter them. Take note, the green color is the process name. Number one, this is called ultrafiltration because I'm filtering all of them out into my Bowman's capsule. Okay, this is like a toilet bowl. That's why I call it Bowman's capsule. Alright, so all the things you have filtered out, we call it glomerular filtrate. Not everything we want to throw it away. Next, we will do second step, which is your reabsorption. We need to take a lot of things back. For example, at the PCT, your proximal convoluted tubule, they will reabsorb glucose, amino acids, salt and water. At your loop of Henle, reabsorb salt and water. And then at your DCT, distal convoluted tubule, they will reabsorb salt and water. Not only that, they will do step 3, secretion. They will throw extra rubbish inside so that you can throw more rubbish out from the body, which is drugs and alcohol. This is how they detect drugs in your urine. And then they will go to your collecting duct, it will form your urine. Okay, do take note one thing here. These parts up here, they are in your cortex of your kidney, and all these part here would be in your medulla. One more thing, a lot of students have problem memorizing your PCT and DCT. This is really simple. Proximal means near. In your MS, you have approximate, means very close value. Distal means distance, means it's very far away. What is convoluted tubule? Convoluted means twisting. Convoluted tubule means twisting tube. So we have your near twisting tube and far twisting. They can't name it that way because that wouldn't sound very nice. Alright, moving on. So you have your negative feedback mechanism, means this is how they do your homeostasis. You have normal value. When it goes too high or too low, they will do corrective mechanism and then they will bring it back. This is what we call negative feedback, and then you bring it into normal value. So I already summarized all the corrective mechanism for you. Very simple to memorize here. Blood glucose, if it is too high, your pancreas will secrete insulin, convert glucose to glycogen, bring it back down, normal value. Too low, release glucagon, convert glycogen to glucose, bring it back up. Osmotic pressure, this is when you don't drink water, you have too high osmotic pressure. Think of it this way, osmotic pressure means thirst. When you don't drink water, your thirst is high, your osmotic pressure is high. So your pituitary gland will release ADH. ADH is to make DCT and CD, which is the last two parts in your nephron, to be more permeable to water, so that more water can be reabsorbed. It makes your urine low volume and very thick, very yellow. When you drink a lot of water, your thirst is low. That means too low. So your adrenal gland, they will release, kindly change something here. This would be your ADH, my mistake here. Let's not talk about aldosterone. Aldosterone is a reabsorbed salt. I'm trying to keep this problem simple here. So your DCT and CD is less permeable to water, less water is reabsorbed, making your urine high in volume and very pale and white. Next, your body temperature. When you are very hot, when your body temperature is very high, you do vasodilation, means your blood vessel will relax to be closer to the skin to lose more heat via radiation. And then your sweat gland will produce sweat, because when sweat evaporates, you have a specific latent heat of vaporization. This is how they carry heat away. You don't lose heat by sweating, you lose heat when the sweat evaporates. Next, you have your erector muscle relax so that your hair goes down, so that they don't trap a layer of air, because you know air is an insulator of heat. This is why when you wear a jacket with fur inside, they trap air, you feel hot. So they don't do that on hot day, so they relax their hair. And then your skeletal muscle, they don't shiver. Adrenal gland, thyroid gland, they are less stimulated, so your metabolism reduced. That's why you feel tired on a hot day. How about when it's too low? When you are too cold, you do vasoconstriction. This is to reduce blood closer to your skin so that you don't lose heat. Sweat gland doesn't produce sweat, obviously you don't sweat in cold. Erector muscle, they contract, means they pull your hair up to trap air, to act as an insulator of heat. Skeletal muscle, they will shiver. This is to generate heat. Adrenal gland, thyroid gland, they are more stimulated, your metabolic rate will increase. This is why you feel hungrier faster in colder climates. Makes a lot of sense, right? Moving on, your plants, you have your auxin here. Remember, auxin will promote growth in shoot, but it will inhibit growth in roots. This is why you see a lot of vaccine here. This part will grow longer in shoot, but a lot of vaccine here in root, it will grow slower. This is why root will bend down. This is what we call positive phototropism and positive geotropism. And of course, you have your ethylene. This is released by ripening fruit. They will cause your food to ripen. They will break down your complex carb into simple sugar. This is why unripe fruit is really sour and ripe fruit is really sweet, because you have simple sugar inside. Next, reproduction and growth. This is one of the most headache chapter. Okay, you have your spermatogenesis and oogenesis. If you look at them, they're really similar. Germinal epithelial cell. Can you see 2N here? It means they are diploid. They will grow into spermatogonia or oogonia. Diploid again. And then they will do mitosis. Why do mitosis here? This is to increase their number. So humans will increase their number first, and then they will go into primary spermatocyte or primary oocyte. Notice the skin outside is called primary follicle. It will develop into a secondary follicle. And then you can see it turns into N here. This is where meiosis 1 happens. This is where meiosis 2 happens. Same thing here, meiosis 1, meiosis 2, and then you have your secondary oocyte, your Graafian follicle. If it is not fertilized, we don't call it ovum. Only when it is fertilized by a sperm, we call it an ovum. So technically, you don't release ovum every month, you release secondary oocyte. Next, you have your ovulation. This Graafian follicle will turn into your corpus luteum. This is the guy that releases progesterone to make pregnancy successful, right? And then you have the spermatid and sperm, and then you have the ovum only if fertilization happens. So your secondary oocyte, they will stop at metaphase 2 every month, waiting for the sperm to come. Moving on, this is how your menstrual cycle works. I'm just going to keep this simple here. Number 1, your pituitary gland every month from around day 1 to day 7, they release FSH. This is to tell your ovary to stimulate it to develop follicle. And then your ovary will reply your pituitary gland saying, okay dude, I got your message. By secreting estrogen, they will inhibit your FSH, telling pituitary gland to stop producing FSH. This is to repair your uterus, getting it ready for pregnancy, for implantation. Next, your pituitary gland will write another letter, another message, which is called LH. This is to stimulate your ovary to release the secondary oocyte. We call it ovulation. Again, your ovary will reply with another message saying, okay, I got your message, which is the 4th message, the progesterone, telling it to stop LH, and this is to further thicken your uterus. Can you see this is sort of like a message and reply thing? After fertilization, you get your zygote. This is the 1-cell thing, and then you have a mass of cell called morula, and then you have a bigger mass of cell called blastocyst, and then you have an implantation. Where's your embryo? These two are actually called your embryo. Embryo is actually a number of stages. Instead of just one stage, we call it embryo. So, identical twin and non-identical twin, what is the difference? 1 sperm and 1 ovum, 2 ovum and 2 sperm. Basically, non-identical twin is when you do 2 pregnancy once, but identical twin is when you do 1 pregnancy, somehow they just separate into 2 fetus. Alright, so you have 1 placenta and 2 placentas, physically similar and physically not similar. Identical twin, for sure they have the same sex, but non-identical twin, they can be same or different sex, but they just look different. One more point, very important, mothers and fetus blood, they do not mix. Why? Because this is to prevent mixing of blood group. Okay, mummies and fetus blood could be different. They could cause agglutination, which could be a very huge problem. High pressure of mother's blood do not damage fetus blood vessel because fetus is still very young. If you have the large pressure going in, you can damage the blood vessel. Harmful bacteria and chemical doesn't enter fetus, but certain chemical can still pass through the placenta going into the fetus. For example, your carbon monoxide, for example, alcohol. This is why it is not recommended for mothers to drink and smoke. Very important in the K-BART question. Quick reminder here, umbilical artery which carries blood to the fetus, they carry deoxygenated blood. Vein will carry oxygenated blood. So in fetus is reversed. So that when mummy's artery has oxygen, fetus artery has no oxygen, only can diffusion happen. Next, contraceptive method. If you don't want baby, this is what you do. Male, you can use condom. Withdrawal method, not reliable at all. And then of course vasectomy, this is sort of like a permanent thing. You just cut away your sperm duct. Not really permanent though, you can attach them back. Okay, female, you have more methods here. You have diaphragm and spermicide. Why do I group them together? You need to use both to make these useful. If you just use either one of them alone, it's not gonna work. And then your female condom, not very common. IUD is the T-shaped thing put in the uterus. This is to stop implantation. And then contraceptive pill and implants, these are basically hormone pills. Basically, it's made from estrogen and progesterone. It will inhibit your FSH and LH. Without FSH and LH, there will be no follicle development and no ovulation. And of course, ligation, this is to cut away your fallopian tube. Without fallopian tube, of course, nothing is going to happen. So, overcoming infertility, if you want baby, on the other hand, what should you do? You can do artificial insemination. Means you do fertilization on a petri dish. And sperm bank, this is to get sperm if you don't want a husband, you want a baby. And surrogate motherhood means you borrow someone's uterus to have a baby. And of course, IVF, this is the test tube baby method. You do artificial insemination outside and then you reinsert it back to the mother. Very expensive process. Okay? This is up to 15,000 to do an IVF. Alright. But hey, if you want a baby and if you can't have it, this is the only way to do it. So plant is different. Just now humans, we do mitosis first and then we do meiosis. Plant is different. They will do meiosis first, only they will do mitosis. So in males, you have pollen sac mother cell. Females, you have embryo sac mother cell. They will become 4 microspore and 4 megaspore. Remember, males one are always smaller. Sperm is very small, that's why we say microspore. Ovum is very big, that's why we call it megaspore. So we only use 1 megaspore, the other 3 will degenerate. So this 4 microspore, each microspore you have 1 tube nucleus, 1 generative nucleus. So this tube nucleus will form your pollen tube. Your generative nucleus will form 2 male nuclei. Why 2 male nuclei? Because you have your double fertilization. So your megaspore will do 3 mitosis to form 8 nuclei, and then they will split into 3 groups. You have your 3 antipodal cells up there, 2 polar nuclei in the middle, 2 synergic cells at the bottom, and then you have 1 egg cell in the middle. So double fertilization, your 1 male nuclei, remember you have 2 male nuclei, 1 will fuse with the egg cell to form zygote and embryo. This is your baby plant. And your 1 male nuclei will fuse with your polar nuclei to form triploid nucleus. This is to form endosperm. Endosperm is the food storage for plant, don't forget. And of course, the ovary will become the fruit. When you are eating fruit, you are eating ovary of a plant. Growth, you have 3 zones: zone of cell division, zone of cell elongation, zone of cell differentiation, and mature tissue part. This is why veggies, they taste the best up here, because you are eating young cells. This is where everything feels hard and not nice to chew, because it's all old cells. Alright, so you have your sigmoid curve in human and this step curve in insect. Human, you have a rapid growth phase during infancy, slow growth during childhood, which is around 3 to 7 years old. And then when you hit adolescence, that's puberty, you have your rapid growth phase. Remember, female will hit puberty first. This is why at around 12 they grow taller than males, and then males will catch up at around 13 to 14. Alright, so females will end their puberty faster, males will end it slower. Female will stop around 17, males will stop around 21. So adulthood, there will be no growth here. Old age, this is where you have negative growth because you drop a little bit in height because the cell starts to die. In insects, you have something called your ecdysis. All these vertical lines are ecdysis, means they inhale air and they will break the exoskeleton and form another new exoskeleton. And at these horizontal lines, where no growth happens, these are what we call instar stage. So we have instar, ecdysis, instar, ecdysis, instar, ecdysis. They will keep doing it until they reach their genetic potential size. Next, you have your chapter 5 and chapter 6. I put these two together because they are very similar. So Mendel's first law is what we call law of segregation. Law of segregation involves only one trait, which we call it mono-hybrid cross. Mono means we only look at one characteristic. So this is the first experiment he did. He used a pure breed tall plant and a pure breed short plant. They crossed them together. By the way, if you have 2 big T, this is what we call homozygous dominant. Homo means same. And then we have, when you produce this big small T, this is what we call heterozygous dominant, which means is different. Hetero means different. This one, of course, this is called homozygous recessive, right? So this is when they cross. Please learn how to draw this diagram. This carries 4 marks in your exam. Alright, so make sure you know how to label. Your phenotype means something that you can see. Genotype means the gene. And then you have your gamete here. You have your offspring genotype and your offspring phenotype. And then Mendel crossed these two, the F1 generation again. And then he got this tall, dwarf, which is the ratio of 3 to 1. This is Mendel's first law ratio. And then Mendel's second law is the law of independent assortment. Now he looked at 2 characteristics, which is what we call dihybrid cross. So you should learn how to draw this Punnett square here. You have 16 results. Big R being dominant means they will be round. Big Y means they will be yellow, which means dominant. And of course, wrinkle is recessive. Green is recessive. Really straightforward stuff. Moving on, you have your blood group. Okay, I drew an example here. When your father is AB and your mother is O, AB is the dominant. That's why we call it co-dominant here. O is the recessive. When an AB person marries an O person, you have 50% of getting an A group child and of course another 50% of getting a B. Rhesus factor is when, okay, if someone is rhesus negative, means positive can harm them. So I already include the explanation here. So if a woman is rhesus negative here and her child is rhesus positive, you have a problem. So first pregnancy of rhesus positive baby, it will trigger the mother's immune system to produce rhesus antibody to attack the fetus. But before the baby can be harmed, he or she would have already been born. And then when second pregnancy happens, if the baby is again rhesus positive, now you have another problem, because during second pregnancy, the mother's immune system now is ready to attack the baby. This could cause anemia and brain damage. Alright, moving on. So human chromosome is 46, 44 autosome, which determines all your characteristics like your eye color, your hair texture, your skin color, and so on. And of course, the last two is what we call your sex chromosome. XX is female, XY is male. Down syndrome is when you have 3 chromosomes like chromosome number 21. This is why Down syndrome is also sometimes called trisomy 21. Tri means 3 chromosomes at the chromosome number 21. Next, sex-linked disease means the disease carried only at the sex chromosome. For example, you have your hemophilia, means they cannot clot blood. So male, you have only either a normal male, means they have dominant gene, or a hemophiliac male, means they have a recessive gene. Female is a bit more tricky because it can be big H, means they are normal, small h means they have the disease, they are hemophiliac, or they could be big small, means this female is normal but they are carriers, means they have the gene, they could pass this to their child and the child might have hemophilia. This can be applied for color blindness as well, so it's the same concept. If you have a normal father marrying a carrier mother, so this is how problem can arise. So you can have a normal daughter, or you can have a carrier daughter, or you can have a normal son or a hemophiliac son. If they know that the pregnancy is a male, is a boy, 50-50 chance he could have hemophilia. So I think you can read this diagram yourself. Moving on, you have continuous variation and discontinuous variation. This is the easiest part to score. I'm sure you learn your AdMaths. Continuous variation is when you have this bell-shaped curve, so they are not distinct, means you cannot separate them into class. Discontinuous, you can separate them into classes. Alright, so factors: continuous is affected by genetic and environment. Discontinuous is only affected by genetic. And inheritance, no, you cannot inherit continuous variation. But discontinuous, yes, it is fully inherited. Intermediate values, yes, you have intermediate value, no intermediate value. Type of data is quantitative, means it's about numbers, and qualitative means it's about yes or no. Alright, so for example, height. Okay, height, you have no distinct value. You could be 171, 172, 173, 174, and so on. Okay, every value has individuals. Okay, same goes to body weight and skin color. You have a range of values rather than a set value. But blood group, you have only 4 types. Type of fingerprint, you have only 4 patterns. Ability to roll the tongue is either a yes or a no. So you have very distinct classes here. Okay, so type of mutation, you have chromosomal mutation, which is a very serious mutation, and gene mutation, which is a less serious mutation. So gene mutation is when you have a gene at fault which causes things like sickle cell anemia and albinism. So your color blindness and hemophilia is also a gene mutation. You will be normal, it's just that you have certain problems. But chromosomal mutation is not so simple, because you have one whole chromosome missing or you have one extra chromosome. This is a huge problem. So it changes the number or the structure of a chromosome. For example, you have Turner syndrome, which means you are a female with one less X chromosome, or you could be a Klinefelter syndrome, means you are a male with one extra X. So these individuals are usually infertile and they are usually mentally retarded. And of course, Down syndrome. One more thing I think I missed here is your homologous chromosome here. I'll just write HC here. So when you have 2 genes here, these are what we call alleles. Okay, think parallel. So this is your dominant gene, this is your recessive gene. And the place that they are located at, this is what we call locus. Okay, location, locus. Okay, these are the few words that you need to know. And of course, phenotype and genotype. Okay, so that's it for this video. Again, I know it sounds a bit rushed, but if you get these 80% down as the last video, more than enough for you to get A. Alright, so I hope this video is helpful. And I know it's only 1 or 2 more days to your biology paper. I'm sure you are nervous. All I can say is make sure you get enough sleep. Please don't do last minute study. It doesn't work. get yourself in a correct state of mind, get rid of any negative thoughts, get rid of any past bad results that you might have. So I hereby wish all of you the best of luck. Okay, selamat maju jaya, and I hope that you come out at the other end with the result that you want. I will see you again soon. Good luck and